inside this room. The furniture, the floor, the rug, everything in sight is made up of chemicals. In fact, so are we. The tissues, cells, and fluids of our bodies are all composed of different chemicals. Yet, in important ways, these chemicals are different from those in a curtain or a chair. For one thing, the chemical reactions in a chair, if they take place at all, occur so slowly that years from now the chair will probably look the same. The chemicals in the human body, on the other hand, undergo billions of reactions every second. How does this happen? Human beings, along with all living things, have enzymes that facilitate chemical reactions. In fact, if it were not for enzymes, we wouldn't have the muscular energy to turn a page, or the nervous energy to read a sentence. Indeed, without enzymes, life itself would be impossible.
Let us take another look at the chair. What prevents the molecules in a chair from reacting? The answer is that these molecules need to be pushed into reacting. The molecules of an inanimate object, such as a chair, face a situation similar to that of a skier at the bottom of a steep hill. In order to have a good downhill coast, she'll first have to spend a great deal of energy getting to the top of the hill. Once at the crest, she can relax and look forward to a long, almost effortless downhill ride. Similarly, in order to react chemically, molecules must first be provided with some energy to become activated. In a way, they must surmount an energy hill, known as the energy of activation. This hill can be thought of as a barrier. Once at the top, the molecule has crossed the barrier and is free to react and form different molecules called products. But how can molecules be brought to these energy peaks where they're capable of reacting? One way is to heat the molecules up. Heat causes molecules to move around faster and bump into each other. And the harder and more frequently molecules collide, the more they react. When molecules react, chemical links called bonds are either formed or broken. By applying intense heat to the wood in a chair, for example, we would cause molecules to speed around, break apart, and combine with oxygen in the air. In other words, the chair would burn. But such intense heat would have disastrous effects inside the body. It would kill the body's cells, and life would be impossible. Another way to increase the likelihood that chemicals will react is to increase the concentration of the chemicals involved in the reaction. The concentrated supply of oxygen in wind and rain, for example, causes certain metals to oxidize or rust. But again, this is not how living organisms work. A third way to facilitate chemical reactions is with catalysts. A catalyst is a special substance that speeds up a chemical reaction without itself being affected. In the human body, catalysts, rather than heat or concentration, are used to speed up chemical reactions. Catalysts that do their work in living organisms are called enzymes. Like all catalysts, enzymes work by increasing the likelihood that chemicals will react. It's as if they lower the energy hill chemicals must climb before they can react. An enzyme can't bring about a reaction that would be otherwise impossible. All an enzyme does is make it easier for reactions to take place.
An enzyme is a type of protein. Proteins are large molecules made up of chains of chemicals called amino acids. The sequence of amino acids in these protein chains determines what protein it is. The largest enzymes are made up of many long interwoven chains containing thousands of amino acids. these chemical chains bend and fold into characteristic shapes. An important characteristic of each kind of enzyme is its unique architecture resulting from the pattern of these folding protein chains. The substances enzymes act on are called substrates. The unique shape of an enzyme allows it to fit closely with a particular kind of substrate, somewhat in the fashion of pieces of a jigsaw puzzle. When the substrate and enzyme combine temporarily, they form an enzyme-substrate complex. The substrate joins the enzyme at a particular place on the enzyme surface called the catalytic site, or active site. The shape of this site and the chemical nature of the amino acids in the site determine which substrate is welcome and which ones are not. The enzyme lowers the amount of energy needed to start a chemical reaction. This reaction leads to the liberating of substances called products. After the products are liberated, the enzyme is free to attach to another substrate. The fit between a substrate and an enzyme is so precise that most enzymes can distinguish among the thousands of compounds in the body and combine with only one particular substrate. Stated another way, for any enzyme, there's usually only one substrate that will fit in its catalytic site.
There are over 1,000 kinds of enzymes at work in the human body. In fact, there is generally one kind for each different chemical reaction that takes place in the body. Some enzymes are specific as to where in the body they do their work. For example, enzymes that help our nerves to transmit impulses or that enable our eyes to see work within nerve or receptor cells. Enzymes that catalyze reactions that enable muscles to contract are found in muscle cells. Some enzymes, however, are found in all the body's cells. Some of these enzymes, for example, control the reactions that give cells life-supporting energy. There are also enzymes, such as those that help digest food, that do not operate within cells at all, but are part of the fluids secreted by the body's glands.
Let's examine more closely how enzymes secreted by different parts of the digestive system operate. Digestion is a multi-step process through which the proteins, carbohydrates, and fats in our diet are broken down into simpler compounds that can be used by the body's cells. It is a process that can begin in the mouth the moment we bite into a piece of food. Glands in the mouth secrete saliva, which contains the enzyme salivary amylase. This enzyme begins the process of breaking down starches, a kind of carbohydrate, in foods like pasta and bread. Other aspects of digestion take place further down the digestive tract in the stomach. Here, for instance, the enzyme pepsin begins the job of breaking down the proteins in such foods as cheese, meat, and fish. After the stomach, food particles travel to the small intestine, a 20-foot tube coiled in the abdomen. A nearby gland, the pancreas, secretes a number of enzyme-containing fluids into the small intestine to aid in digestion. For example, the enzyme pancreatic amylase continues the process of breaking down starches. The enzymes trypsin and chymotrypsin take particles of protein partly digested by pepsin and break them down further. And fats are broken down by enzymes called lipases. Altogether, there are dozens of different enzymes involved in digestion, a few of which are shown on the screen. Each one of these enzymes performs a specific task. The ability of enzymes to break down compounds in food has been put to a number of commercial uses. For example, cheesemakers use rennin, an enzyme from the stomach of a calf, to curdle milk. The enzyme papain in the papaya fruit can split proteins, so it's used as the active ingredient in commercial meat tenderizers. And some detergents contain enzymes that help remove stains by breaking up proteins.
In breaking down molecules such as those in food, enzymes don't operate alone. Water is also involved through a process called hydrolysis. For example, milk contains a double sugar called lactose that consists of two simple sugars, glucose and galactose. The enzyme that breaks down lactose into its simple sugars is called lactase. In the presence of water, the enzyme lactase splits lactose into two products, glucose and galactose. These products themselves may break down further in a series of steps involving other enzymes. Glucose is particularly important because it is an energy-rich compound that is the body's major fuel. Before the energy in glucose can be used, however, glucose must first undergo a series of chemical reactions in the cell. This process is called cellular respiration, and it requires many different enzymes. For the most part, cellular respiration takes place in bodies in the cell called mitochondria. The inside of a mitochondrion is organized into long inward-folding folds that provide an efficient space for enzymes to work. It is here that the chemical reactions that provide most of a cell's energy occur. In assembly line fashion, series of different enzymes perform different steps in a process whose final result is the formation of molecules of adenosine triphosphate, or ATP. It is ATP that ultimately provides cells with energy.
To understand how ATP works, it is necessary to examine its chemical structure. ATP consists of 1 unit of adenosine and 3 units of phosphate molecules. The chemical bond that attaches the last phosphate molecule is rich in energy. An enzyme called ATPase catalyzes a reaction through which this bond is broken, removing the third phosphate. As a result, energy is released. It is ATP that provides the energy for all the cell's activities and permits the body to function at all. The energy released by ATP powers the muscles in the arms of this kayaker, or the nerve impulses that flow throughout our brains as we write a poem or form a thought. But without enzymes such as lactase, ATPase, and all the enzymes involved in cellular respiration, the chemical reactions necessary for energy would occur far too slowly. We wouldn't have enough energy to lift a finger. Indeed, life itself would be impossible.
So far in this program, we have discussed how enzymes can speed up chemical reactions in which substrates are broken down into products. We have seen how the reactions are important for, among other things, digestion and the release of energy. But another kind of reaction is also possible. These reactions start with 2 or more substrates. And with the help of an enzyme, these substrates react with each other to form a single product. Such reactions are known as synthesis reactions. Like reactions involving digestion and energy, synthesis reactions are crucial for our well-being. This is because cells synthesize all the materials that make up the body, the cell components, blood, hormones, enzymes, proteins. These and other body materials are the result of chemical reactions regulated by enzymes. The growth of a child is only one example of the importance of synthesis reactions. The bones, tissues, and nerve cells that form as a child matures into an adult all result from reactions made possible by enzymes. Because enzymes are so important to the body's chemistry, they have long been the focus of intensive research. One result has been that scientists have somewhat revised their views about how enzymes operate. It was long believed that an enzyme and its substrate fit together perfectly, in much the same way as a lock and key. Indeed, for the sake of simplicity, this is the model that we generally use throughout this program. However, recent research indicates that while the enzyme and substrate do fit closely,
they don't join with lock and key precision. Their shapes are slightly different and require a bit of molding to fit. This model of how a substrate joins with an enzyme is known as the induced fit model. For this model, a strain is put on the substrate as it changes its shape to fit into the enzyme's catalytic site. Biologists believe this strain on the substrate may weaken the bonds to be broken, lowering the substrate's resistance to reacting. Finally, the bond is broken. In this example, what was once a substrate is now two products. Its work done, the enzyme snaps back into its normal shape, ready for the next substrates to come along. One single enzyme may go through this whole process with thousands of substrate molecules a second.
Understanding the speed of enzymes is particularly important in appreciating how they work. For example, one of the body's fastest-acting enzymes, catalase, is found in a variety of human cells. It catalyzes the breakdown of hydrogen peroxide to water and oxygen. Hydrogen peroxide is a common byproduct of many natural chemical reactions within the human body. But there's a problem. Hydrogen peroxide is poisonous to cells. However, in 1 minute, a single catalase molecule can split 5 million hydrogen peroxide molecules to form water and oxygen. This is 1 trillion times faster than the rate at which the reaction would occur without an enzyme.
The example of hydrogen peroxide confirms the importance of enzymes to the body's health. In this part of the program, we have seen some of the ways in which enzymes serve the body. Enzymes are necessary for the energy that powers all the body's activities. Enzymes help to digest the food we eat so that it can be used by our body's cells. And because of enzymes, cells are able to manufacture the materials the body needs to maintain itself and grow. But enzymes don't always work flawlessly, and when they don't, the body's health is threatened. Part 2 of this program will explore in greater depth how enzymes contribute to our health and what happens when things go wrong.
Stephanie is an albino. She was born with delicate hair that's more white than blonde. Her pale skin is so sensitive that prolonged exposure to the sun would jeopardize her health. The cause of Stephanie's condition is a single missing enzyme that is necessary for the body to produce melanin, a pigment that colors hair, skin, and eyes. While albinism is not a life-threatening condition, other enzyme-related disorders are more serious. This presentation will examine how enzymes work to ensure our health and well-being, and what happens when they don't work well.
In the body, enzymes operate in much the same way as the assembly line in a car factory. In the beginning of the automobile era, Henry Ford recognized that it would be inefficient for just one individual to build a whole car. Ford organized his factory so that many laborers worked side by side, each doing a specific job. For example, one worker might be responsible for tightening a specific bolt, while another would attach a tire. In this way, a finished car was the product of a long series of steps involving many different workers. In a similar way, the chemical reactions in the body take place in an ordered series of steps, each step involving an enzyme that performs a specific job. These series are called enzyme pathways, and different pathways serve such vital functions as providing energy for cells, digesting food, and synthesizing new body tissue. Like the workers on an assembly line, the enzymes in a pathway function in a variety of ways. Some enzymes may split up molecules into smaller products. Other enzymes may do no more than clip a hydrogen atom off their substrate. And still others may accept two substrates, taking a chunk from one and transferring it to the other.
This greatly simplified diagram illustrates how an enzyme pathway as a whole might work. A molecule is changed a little bit by the first enzyme, a little more at the second, and so on until the end product is produced. But what happens if an enzyme is missing from a pathway? The result is that the pathway is disrupted, and the effects depend on the particular enzyme and the particular pathway involved. One example might be the enzyme tyrosinase. When this enzyme is missing, the body fails to produce the pigment melanin. The consequence, as we've seen, is an albino, like Stephanie. Tyrosinase is ordinarily part of a pathway that controls several activities in the body. More serious consequences result if the enzyme right before tyrosinase in the same pathway is missing. This enzyme is called phenylalanine hydroxylase. When phenylalanine hydroxylase is missing, the result is a disease called phenylketonuria, or PKU. PKU can lead to severe mental retardation and often results in an early death. Fortunately, a special diet started very early in life can prevent these consequences. PKU is a hereditary disease, the result of a pair of defective genes passed down to a child from his or her parents. Genes determine how hereditary information is transmitted from one generation to the next. They are crucial for survival because they regulate the manufacture of proteins, the body's building blocks. Since enzymes are proteins, the kinds of enzymes manufactured in the body are dependent on specific genes. Consequently, a defective gene or genes can mean that not enough of a particular enzyme, or no enzyme at all, is produced. This deficient or missing enzyme, in turn, can cause the breakdown of an enzyme pathway that is responsible for important chemical reactions in the body. So genes play a critical role in how enzymes affect our health.
Other factors besides genes can also be important to how enzymes work. Up to this point, we've been viewing enzyme reactions as involving two primary participants, enzymes and substrates. The fact is, however, that a great many enzymes cannot catalyze reactions by themselves. Sometimes, for example, an enzyme and a substrate don't fit together properly. In this case, an additional molecule is required, one that provides something the enzyme needs in order to react with a substrate. Such molecules are called cofactors. Many enzyme-catalyzed reactions would be impossible without them. One important group of cofactors is minerals, such as magnesium, iron, calcium, and zinc. These minerals can be purchased as dietary supplements. But our primary source of minerals is the food we eat every day. Vegetables, such as tomatoes, beets, and peppers, are rich in minerals that are essential for the proper functioning of certain enzymes. Spinach is another food renowned for the mineral content. When we eat spinach, digestive juices in the stomach break it down and release the minerals. Among these free minerals is the metal magnesium. Free magnesium atoms find their way to the catalytic sites of certain enzymes. Here they form temporary chemical bonds with their host enzymes. With its magnesium cofactor in place, the enzyme can attract its substrate and make it easier for a chemical reaction to occur. Magnesium is essential for many of the important chemical reactions in the body. For example, the synthesis of any protein in the human body cannot take place without magnesium as a cofactor. Examples of other minerals vital to our health are calcium and iron. Calcium plays a role in reactions that enable the body to contract muscles and conduct nerve impulses, while iron is necessary for enzymes that help ensure that the body's cells get enough oxygen.
Besides minerals, there are other types of cofactors that are vital to our health. These are called coenzymes. Unlike enzymes, coenzymes and other cofactors are not proteins. But coenzymes differ from minerals in that they are complex organic compounds. Some vitamins play coenzyme roles and are essential participants in a number of chemical reactions. Like other cofactors, coenzymes help enzymes and substrates bind to one another. While everyone is familiar with vitamin supplements, eating the right combination of foods easily provides all the vitamins we need because the body requires vitamins in very small amounts. If a diet is not well balanced, however, a deficiency in vitamins can result in serious diseases. In the past, for example, sailors on extended voyages suffered from scurvy, a disease that results from a diet deficient in vitamin C. But an English surgeon demonstrated that by drinking a lot of lemon or lime juice, both rich in vitamin C, scurvy could be cured or prevented from occurring at all. British sailors, in fact, were required to drink so much of these juices that they were soon nicknamed limeys. Peanuts are a good source for another important vitamin, niacin, a member of the B vitamin family. As niacin is digested, it changes form slightly and joins with other chemicals to form a substance called nicotinamide adenine dinucleotide, or NAD for short. NAD is involved in most of the enzyme reactions that release energy for our cells, particularly skin, nerve, and digestive tract tissue. What happens when niacin is in short supply in the body? Some of the symptoms are sleeplessness, nervous irritability, headaches, and stomach aches. They occur because without niacin, many enzymes are deprived of the coenzyme, without which they cannot do their work.
We've seen how cofactors such as vitamins and minerals contribute to our health by helping enzymes do their work. But there is another class of substances that prevent enzymes from doing what they're supposed to do, or change the way they act. These substances are referred to as inhibitors. Some inhibitors are competitive, so-called because they compete with substrates. A competitive inhibitor looks very much like an enzyme substrate and can fool the enzyme into binding to it rather than to its substrate. But the enzyme is capable of catalyzing only a reaction involving its true substrate. It can't do anything with the false one. A competitive inhibitor, in effect, poisons the enzyme, putting it out of commission. Scientists have learned how to put inhibitors to constructive uses, destroying harmful bacteria. In fact, one day in 1928, some inhibitors wreaked havoc with a whole colony of bacteria that were part of an experiment conducted by biologist Alexander Fleming. Fleming had forgotten to put the lid back onto the dish where his bacteria were growing. A mold spore had floated in through the window and had landed in the dish where it grew and killed Fleming's bacteria, ruining his experiment. Many other scientists would have pitched the moldy mess into the trash. But Fleming studied the mold and its deadly action on his bacteria. After many years of investigation, it was shown that a substance in the mold, penicillin, acted as an inhibitor. By attaching itself to the bacteria's enzymes, the penicillin prevented them from combining with their true substrates, and so the bacteria died. The discovery of penicillin was a major breakthrough in the field of medicine. Since the 1940s, penicillin and other bacteria-killing substances, called antibiotics, have been used to cure many diseases.
Scientists have used their growing understanding of how enzymes work to develop other types of drugs as well. An example is SULFA drugs. Like penicillin, SULFA drugs work by competitively inhibiting enzymes necessary for bacteria to live. Here is how SULFA drugs work. An enzyme on one of the bacterium's enzyme pathways combines with a substrate called para-aminobenzoic acid, or PABA for short. One of the products of this enzyme pathway is folic acid. Folic acid is essential for any form of life. Some SULFA drugs look so much like PABA that whenever bacteria are exposed to them, they trick the bacteria's enzymes into uniting with the drug instead of with PABA. This inhibited enzyme short-circuits the entire pathway, so folic acid can't be synthesized. Unfortunately for the bacteria, they die. Since humans need folic acid, why aren't they poisoned by SULFA drugs? The answer: humans get folic acid directly from their diet. Since the body has no need of an enzyme to aid in synthesizing PABA, there is no enzyme for SULFA drugs to inhibit.
Competitive inhibitors such as antibiotics and SULFA drugs act by binding to an enzyme's active site, preventing the enzyme from combining with its true substrate. Some inhibitors, however, are non-competitive. That is, they bear no resemblance to an enzyme substrate. And instead of fitting onto an enzyme's active site, a non-competitive inhibitor attaches itself to another part of the enzyme. But when a non-competitive inhibitor attaches itself to an enzyme, it changes the enzyme structure so that it cannot interact normally with its substrate. Here, for example, the enzyme's active site has been widened. Because they prevent enzymes from functioning normally with their substrates, some non-competitive inhibitors, such as cyanide and mercury, can be highly poisonous. Lead is another example of a poisonous non-competitive inhibitor. Up until the 1950s, lead was a common ingredient of interior house paint. Curious but unwary toddlers would pick chips of old paint off the wall and eat them. Inside the body, the lead from the paint would attach itself to certain enzymes, rendering them useless, unable to combine with their substrates. The results? Weakness, weight loss, and nausea. In severe cases, lead poisoning was found to lead to convulsions, retarded growth, and even death. For these reasons, there are now laws that forbid the use of lead-based paints on children's toys and in house paints.
While some non-competitive inhibitors are poisonous, others serve a useful function by helping to regulate how enzymes work in the body and making sure they work efficiently. They do this through a process called negative feedback. Here is how negative feedback operates with enzymes. Earlier, we saw how enzymes work in pathways to synthesize new products for the body. Each enzyme affects one specific chemical reaction in a cell, and in the end, a product is formed that the cell needs. But when too much of this product is produced, it returns to the first enzyme in the pathway through a feedback loop. Here, it acts as a non-competitive inhibitor. Almost as if biting the hand that fed it, the end product attaches to the first enzyme, altering the enzyme's shape and thereby preventing it from reacting normally with its substrate. The result is that the entire enzyme pathway is shut down and no more end products can be formed. This is one way the body protects itself from producing too much of certain substances. But what happens when the body needs more of the end product? In this situation, the end product separates from the first enzyme, freeing the enzyme to bind to its natural substrate. The entire enzyme pathway is once again set in motion to produce more end products.
Another factor affecting enzymes is the acidity or alkalinity of their environment. Most enzymes in the human body work best in a neutral environment, one that's neither acidic nor alkaline. There are, however, major exceptions to this rule. The intestine happens to be quite alkaline, and the digestive enzymes trypsin and chymotrypsin work well here. The stomach, on the other hand, is acidic. This environment suits the enzyme pepsin. Temperature is another environmental condition that affects enzymes. Normally, the optimal range for enzymes in the human body is between 20 and 40 degrees Celsius, or 68 and 104 degrees Fahrenheit. However, some enzymes function efficiently in a very cold environment. For example, the enzymes that regulate the chemical activity in cold water fish, such as this cod. On the other hand, the enzymes that keep alive the microorganisms that live in hot springs operate in extremely hot temperatures. But when enzymes in the human body are subjected to such hot temperatures, they lose their characteristic shape. The process is called denaturation. A denatured enzyme, like an inhibited enzyme, cannot bind to its substrate. When many enzymes are denatured, life is threatened. This is why it's so important to control a high fever. A temperature of 42 degrees Celsius or 108 degrees Fahrenheit almost surely results in death.
When enzymes operate smoothly, most of us pay little attention to these remarkable chemicals. Yet enzymes play an indispensable role in most of the chemical reactions in the body that make life possible. Often, it is only when enzymes don't function properly that people begin to appreciate their importance. Many diseases have now been traced to enzyme-related problems. As medical scientists learn more about enzymes, many enzyme-related disorders can be treated. And scientists can also use enzymes to fight various diseases and ailments. As research continues, further breakthroughs may occur. But with or without such breakthroughs, it is increasingly evident that our health and well-being are dependent on the intricate, complex activities of enzymes.